Hey, you can hear me. Good morning, good afternoon, good evening, our fellows. Welcome to today's review session. Just a minute. Okay. Okay, good morning, good afternoon, good evening, our fellows. Welcome to today's review session. My name is Lavenda Ochieng. I am a research assistant at ARIN, that is Africa Research and Impact Network, and I will be your moderator for today. So just to briefly introduce our topic and the speaker, and I hope, Gerald, you are able to share your screen. If not, you can share it with me. I help from my side. So in today's review session, we are going to look at the local feeding practices that can be adopted to mitigate enteric methane in sub-Saharan livestock. So we all know that in sub-Saharan, livestock plays a critical role in both livelihoods, in food security, and even in economic development. And these livestock, especially the ruminant animals, such as the cattle, the sheep, the goats, they produce an enteric methane that is a potential greenhouse gas. And this is a great contributor to climate change today. So in response to this, researchers and even scientists are coming up with innovative feeding practices that not only sustain productivity, but can also adapt to the impact of climate change. So in our today's review, we'll delve into this topic. We will look at how or what are the various innovative practices that the most people are putting in place or that has been said to be potentially viable in mitigating climate change. And our speaker who is going to take us through this discussion is Mr. Gerard Xavier, who is from Benin. He is a RIN fellow, is a researcher in livestock systems and greenhouse gas assessment at the University of Abomey-Calavi in Benin. Is having a strong interest in adaptation and mitigation on climate change in sub-Saharan Africa. Is passionate about bridging research and policy practices, and additionally collaborates with rural community and livestock farmers to co-develop climate solutions. So Gerald's really doing amazing work out there. And in our today's session, we look at the various, one of his paper, the research that he did, looking at how innovative solutions can be adopted by pastoralist farmers to mitigate climate change. Gerald, if you can hear me, I hand it over to you. Kindly just share the screen and take us through the presentation. Over to you.
Yeah, my screen is already shared. Are you seeing it?
No, we can't see you're actually there. I don't know, click it, maybe.
Okay.
Or you can share it with me. I help you from my side, please, if there is a challenge. Yes, put it in the presentation mode.
It's good now?
Yes, please continue.
Okay.
You can go ahead, Gerard. You can see the screen well.
I can start now?
Yes, go ahead.
Okay. Thank you. Thank you very much. Hello, everyone. Welcome to that presentation titled Local Feeding Practices to Mitigate Enteric Methane Emissions in Sub-Saharan African Livestock. I am Dr. Gerard Xavier D. I'm a livestock system and greenhouse gas assessment specialist. It is important to notice to you that this presentation is a part of my PhD research. The main joints of that presentation are successively contest objectives, methodology, result in discussions, conclusion and advice, perspective. The environmental concerns related to greenhouse gases emission is now at the center of reflection about the development of livestock. In Sub-Saharan Africa, particularly, the livestock system is extensive. And that kind of system is rightly or wrongly regarded as generator of the world's highest yield and intensity of greenhouse gases, particularly enteric methane. But those statements are not supported by accurate data considering local resources, feed seasonality, feed diversity, feed availability, local ruminant breeds that are recognized to influence enteric methane emission. However, several techniques were reported in literature to mitigate this gas. Among those techniques reported, feeding has proved to be the most promising strategies. And those feeding strategies reported are limited at research stage in Sub-Saharan Africa. But why? The main reasons are competition with humans, the cost, the complexity for using, and the lack in generating profit for farmers. It is accepted that farmers use local resources such as crop co-products, agro-industrial byproduct, and tree legume to feed the ruminants. These resources available to farmers should then be explored in the context of enteric methane mitigation. It is why the main objective of that study is to analyze the synergy among intake, digestibility, and enteric methane in Sudanese Fulani Zebu, that is a local cattle breed, according to local feeding strategies in Sub-Saharan Africa. That general objective is declined into three specific objectives. The first is to compare the intake and refusal quality in cattle fed with different local diets. The second is to analyze the diet digestibility. And the third, to compare the emission in cattle fed with those local diets. But what was my methodology adopted? That study was carried out at experimental session in an international center of research on livestock called CIRDES, located at Burkina Faso. This area is under the influence of three different seasons over a year. They are called dry season, hot dry season, and wet season. My study was conducted during the hot dry season. That corresponds to the period when farmers supplement their ruminants because of the short of resources. This study involved 10 Fulani Sudanese Zebu steers, aged 33 months old, and weighing 142 kilograms initial. 2 control diets were used for this study, and 6 crop co-products were used. Why did we use 2 control diets? In the proposal, it was designed to use one control diet, that is the rangeland fodder, as control. But as I said, the study was carried out in the hot dry season. And you know that from the beginning to the end of dry season, the quality of fodder in rangeland decreased drastically. So we started well the study, but toward the end of the season, we were confronted to the missing of rangeland fodder on the pasture. So we replaced the rangeland fodder with a grass, Panicum maximum, harvested at the mature stage, and that has practically the same nutritional quality than rangeland fodder. The 6 crop co-products that we used were parted into 4 cereal co-products and 2 legumes co-products. Those 6 co-products chosen were with the participation of farmers. So the study is divided into 2 experiments. The first experiment is where rangeland fodder is the control diet. And with rangeland fodder, we test the 4 cereal co-products that are sorghum, rice, maize, and millet straws. The second experiment is when Panicum maximum was the control diet. And with this grass, we test coffee and peanut hulls. Each diet was offered to animals at 3.2% of their body weight, dry matter basis. The study lasted 3 weeks, parted into 2 weeks of adaptation and 1 week of data collection per diet and per animal.
National and the
Can I continue?
Yes, sorry about that.
The data we collected was animal initial weights to calculate the quantity of feed to offer to animal each day. We also retired refusal to calculate the intake. We collect the total process per day to calculate digestibility. And we measure enteric methane with green feed system at 6:30 a.m., 10 a.m., 2 p.m., 6 p.m., and 0 a.m. Those periods were fixed according to eating behavior of animals. At 6:30, animals were fasted. 10 a.m. is just after eating because they receive the half of the daily quantity of feed at 8:30. 2 p.m. is the period of rumination. 6 p.m. was just after eating because they received the second half of the daily quantity of feed at 4:30. And 0 a.m. when they are resting. We can see on the slide the bag we fitted to animal to collect total feces. We can also see the green feed system and how the measurements were done. All those data were submitted to a global linear model by ANOVA analysis with fixed FA considered as a diet. What are now the results we reached? This slide shows us the nutrient content of offered diets. At left side, we have the result of experiment 1, and at the right side, we have the result of experiment 2. Each bar here corresponds to one diet. So in experiment 1, we observed that experimental diets that are mixed diets are less fiber than control diets, except diet containing maize straws. But in experiment 2, we observe that mixed diets are not only less fibrous, but also more proteinic than control diet. Sorry, I did not understand what happened. Okay, we were here. Those observations were reported by Jarial in all 2020 when he explained that legume co-product offer high nutritional quality compared to cereal co-product. So the quality of diets depend on the nutritional quality of crop co-product used to mixed rangeland fodder or panicum maximum. That slide shows us the diet intake. In experiment 1, only diet containing rice straws is most ingested than all others diets. It can be explained, but the stocks containing in diet containing maize, sorghum, and millet straws. So it provoked a selection in animal and reduced the intake. In experiment 2, mixed diets are more ingested than the control. Our observation is similar with that reported by Azutan et al. When they supplement ruminate with legume co-product. The selection observed in diets is put in evidence here by the composition of refusal and intake. We can see here that the fiber intake is higher in control diets, but lower in supplemented diets. It is the same things in experiment 2. This slide shows us the diet digestibility. In experiment 1, we observed neutral digestibility in control diets and in diet containing sorghum straw. This neutral pre-protein digestibility can be explained by the endogenous protein synthesis and or the test formation of the intestine that are ejected, excreted by feces. In experiment 2, we observed that crude protein and neutral digestion fiber digestibility are lower in mixed diet than in control. We explain it by the presence of tannin in those diets that provoked the negative effect on crude protein digestibility, as reported by Beshanati in all 2022. That slide shows us enteric methane emissions. In experiment 1, we observed that diets containing sorghum, millet, and rice straws mitigate enteric methane yields by 21, 26, and 23%, respectively. The rate of gross energy intake lost as form of enteric methane is also reduced by 19, 26, and 19% in animals. In experiment 2, enteric methane yield was mitigated by 21 and 19% with diets containing coffee and peanut bulbs, respectively. The rate of gross energy intake lost as form of enteric methane is reduced by 21 and 19% also. The mean of all those values is similar to that of IPCC 2019 and show that IPCC Tier 2 can be used to estimate enteric methane in Sub-Saharan Africa. But when we consider only control diets that are regional fodder and panicle maximum harvested at mature stage, we observed a difference. Our methane yield is very higher, and is a sign that the diversity of diets would be taken into account to improve IPCC Tier 2. The data presented here is published in Archives of Animal Nutrition. We can consult that manuscript to more comprehension. What could we keep in mind from this presentation? This study investigates in vivo enteric methane emission in Sudanese Fulani zebu, that is a local cattle breed according to feeding practices in West Africa. Feeding strategies based on crop co-products improve diet quality. The nutritional quality of feed of cattle ruminants directly influences voluntary intake and digestibility. All feeding strategies tested in this study, except the use of maize straw, resulted in significant mitigation in enteric methane yields. So, raising awareness among agro-pastoralists about the use of crop co-products offer real prospects for this gas mitigation in ruminant from the Sahel region, from the West Africa, and from Sub-Saharan Africa. In perspective, we plan to test other co-products, tree legumes, agro-industrial byproducts used by livestock farmers, because it is not just the crop co-product we tested that farmers used in real environment. We must also take into account the economic aspect, because the first objective of farmers is to make a profit. We will conduct experiments on pasture to perform intake, digestibility, and methane emission across all seasons of the year. We must also conduct similar investigation on other local breeds, because in this study, we just conduct the experiments on Fulani Sudanese Zebu cattle. We will also include taurine made in southern Benin and other kind of Zebu in future experimentation. And the final step of research is to popularize the results. So we must disseminate our result to farmers and help them to apply all the strategies found. This study was made possible by CASES projects. In this project, we continue to explore other feeding strategies to mitigate enteric methane emissions in livestock systems. So now we are conducting experiment on by-products used by farmers, main market garden residues found by pastoralists in pasture, other co-product found after agricultural harvesting, and tree legumes more are concentrated on pasture. I am at the end of my presentation. Your observation, correction, critique, and all recommendations are welcome to improve my presentation and my research. Thank you.
Thank you so much, Gerard, for that presentation. Indeed, this was a really learning platform, and I'm really optimistic that most of us have learned something. Looking at the diet that most farmers or livestock are being fed, or farmers used to feed their livestock, and their digestibility and the reduction rate of the methane that is produced to the atmosphere. So I think we've learned something, and I really just have to follow up with a question, probably. Maybe you may answer this or give us a comment on the same. Looking at the types of diet that you have tackled in your study, ideally they're trying to bring out the aspect of reducing of the gas from the atmosphere, and looking at the type of breed that you looked at in your study. What are the other types of diet that most farmers use today that increases the emission of this gas to the atmosphere, and why are we just advocating for the ones that you've studied? I don't know if the question is making some sense. Question.
Hello?
Hello, did you hear my question?
Hello?
Hello, I can hear you.
You can hear me? Okay.
Yes.
Just resume your question, please. And You are very fast. It is difficult to me to hear because
Okay, thanks. I understand. Yeah, this is the problem of Francophone and Anglophone. We speak too fast. Okay, I understand. So my question is, the study tended to look at the most diet that reduces a methane gas into the atmosphere, right?
Mhm. Yes.
Right. So what are the other products that maybe the farmers currently use that increases this gas into the atmosphere? I just want to understand.
You are asking me if there is a product that will help to reduce the gas, is it?
No, the one you've tackled helps to reduce the gas, right?
How the gas is emitted in environment.
And the result is there's a reduction.
The gas is emitted environment by rot in animal. I don't know if you know rot. By rot.
Maybe it will help you understand. I'll write it in the chat section.
Okay.
Great, thanks. And also, I'll welcome this session to anyone with a comment or a question on the presentation. Kindly just feel free to react or ask Gerald anything. Feel free. Or a comment. Chris, do you have a comment on the presentation?
So you can write your question in the chat.
Yes, I will. So the floor is open to any question. Chris, Emmanuel, in case you have a question. Jim, do you have any question on the presentation? Or a comment as well?
Thanks, Lavender. No question from my side.
A comment. To just give a comment on the presentation.
I'm waiting for your question.
I'll put in the chat section. And there's actually another question from Courier. Dr. Courier, maybe unmute and ask the question.
Oh, okay, thank you very much. Can you hear me?
Yes.
Okay, now that we have the products which are helping in reducing the methane in animals, I have noted like in Kenya, it is a very emotive topic where some people are indicating that there are vaccines which are being introduced to animals to reduce methane. I would like to hear from the presenter on whether in his research he has noted any which is being adopted either in Africa or even worldwide. Thank you very much.
Thank you, Dr. Ari. Gerald, you may answer those two questions, the vaccine and the diet that increases. The vaccine that has been, is there any vaccine which is ready to be adopted to reduce methane in animals?
I can say yes. But in Africa, it will be difficult to use the vaccine, because to allow farmers adopt a vaccine, there are many parameters to be taken into account. The cost, the availability, and the profit it will make for farmers. I wrote a paper that described the most promising strategies to reduce enteric methane. In Africa, it will be difficult to use vaccine. So, because our farmers have another realities than European farmers, for example. In Europe or in Occident, it can be easier to use it, but in Africa, it will be difficult. Not yet vaccine adopted in Africa. I do not think.
Thank you. Thank you for that answer. I hope, Doc, you are well answered.
Yes, that is very okay. Thank you very much.
Thank you too. Thank you too. There's someone speaking. Who is that? Adamu, do you have a question? I can see you are speaking. And maybe you may answer my question then. Which type of other diets do most farmers use that has the potential of increasing this gas in the atmosphere?
It is the most fibrous diets. I conduct another study on the seasonality of rangeland fodder, for example, and we understand that when farmers use grass or rangeland fodder harvested at mature stage, it indulged more methane emission in atmosphere. So it is important to use the green or dried grass, but not the grass already at mature stage. You know, in Africa, it is an extensive system. So even in hot, dry season, farmers are in pasture to feed the animals. So it will be good to supplement the animal in hot, dry season to reduce the amount of enteric methane emit in environment. So the diet used today by farmers and that increase enteric methane emission in atmosphere is forage harvested or directly fed by animal at mature stage.
Okay, thank you for that answer. That is really well understood, and I'm grateful. I don't know if there is another question or a comment before we close this discussion for today. Anyone I'm leaving behind? Okay, I'll welcome Florence just to give us the last closing remark, probably looking at how Irene working on the Llama project can really impact or tap into this project and ensure that the local communities, who are mostly the pastoralists and farmers, get this knowledge and ensure that they adapt to climate change and mitigate CO2 emission or methane emission to the atmosphere from the local level. Flo, over to you.
Thank you very much, Lavender, and many thanks indeed, Gerard, for that insightful presentation. This was a bit technical, but you have endeavored to synthesize it as much as possible. Indeed, Africa is experiencing the severe impacts of climate change, and any practical solution like now this one that helps to mitigate emissions is a step in the right direction. And since methane is a major contributor to climate change, identifying and implementing sustainable livestock feeding strategies is essential for a more climate resilient agriculture. But I think beyond the environmental benefits, these strategies offer significant economic advantages for smallholder farmers, as we may be very much aware that many farmers in Africa struggle with the high cost of commercial feeds, and therefore utilizing the available crop products, as Gerard has said, can reduce feeding costs and improve livestock nutrition without relying on expensive alternatives. And this not only improves livestock productivity, but also supports better incomes and food security for rural communities. But I think also for these solutions to have impact, widespread impact, several challenges need to be addressed. For example, farmers may require greater awareness and training on how to integrate these strategies. And also policymakers and agricultural experts must now collaborate to ensure that research findings translate into actionable practices on the ground. Yeah, so as we move forward, it is important just to explore how these strategies can be adapted to different ecological zones in the continent, taking into account variations in climate, soil conditions, and available crops. As I've always said, this conversation must not stop here. We need more research, more knowledge sharing, and policy interventions just to scale up these innovations for lasting impact. So thank you for joining this week's SARIN review. We hope today's discussion provided insights and inspirations for further actions in making farming in Africa climate friendly and resilient. Thank you, and back to you, Lavender.
Thank you so much, Flo, for those key highlights. Indeed, we, as researchers, we need to be active and proactive in scaling these solutions to the community, and I really appreciate those insights you've put across. I also appreciate Gerald for those findings that you've presented today. We've managed to learn something, and yeah, the ball.
you.
You're welcome. You're welcome. And now it's our the challenge is now on our court, rather to stay with these solutions or ensure that they reach the local communities. And I really appreciate, and most people who have actually joined, I hope you've learned something today. So at this point, I think we'll have to close this meeting earlier today. And maybe, Gerald, you may need to stop sharing the screen so that we take the final group photo.
Okay. Stop
it from my side. So at this point, we'll just take the final group photo, just to show that we had this meeting. Evidence is key in research and every aspect. So maybe I'll request the team to turn on their cameras. And Flo, if you're in a position, just help me take the final group photo. I can see Charles Tonui. Charles, how are you? Florence, will you help us take the final group photo? Just a minute.
I've done, Lavender, we are done.
You could have
Thank you
told
so
us that
much.
we smile. It's fine though. Thank you so much.
People are smiling. Everyone is smiling here.
Okay, great. Thank you so much.
Yeah.
That was really amazing. We appreciate everyone. May you have a fantastic weekend ahead, and let's touch base same time next week. Bye-bye.
Bye-bye.
Bye, Lavender.
Thank you.
Bye bye.
Okay, bye-bye.